Hi guys, how are you? Again, we are looking on the human nutrition. We were talking about the enzymes and we have discussed some of the aspects of the enzymes. These are the things we have covered in previous lectures. So we will be continuing on that, starting with the regulation of the enzymes. The enzymes are regulated. For example, it may be necessary to decrease the activity of certain enzyme if the cell no longer needs the product produced by that enzyme. So there are several ways either that can be regulated through genetic control, genetic regulation. Uh, there is another type of the regulation that is called as the competitive inhibition, non-competitive inhibition, and the feedback inhibition. So all of these methods are used to regulate the enzyme at the cellular level. So in the competitive inhibition process, a similar type of the molecule that competes with the substrate for the active site. So if we look at the active site, so you can see this is the uh, active site. This is the active site. So this is placed at this direction. So look at this one. Originally, this is meant for this substrate, but this is another substrate that is competing for the active sites. For the non-competitive inhibition that works on other than the active site, for example, this is the inhibitor when that is attached with that enzyme that change the configuration of that active site. So the substrate that cannot be actually positioned at that point. So this is the non-competitive type of the inhibition, the binding of an inhibitor to an allosteric site. And normally that is the temporary and based on the requirement of the cells. Poisons that can be acting as the inhibitor bind on those sites irreversibly. Penicillin inhibits an enzyme needed for the bacteria to build the cell wall, and so many examples for that. And the third type of the inhibition process, regulation inhibition process, that is called as the feedback inhibition process, in which some of the product of the reaction that inhibit any type of the enzyme. For example, if the D is the product that is serving as an inhibitor for the enzyme 1, so it's mean at that time all other products that will be inhibited and the working of the enzyme 2 and the enzyme 3 in this case that will be restricted. So look at this one, D that is inhibiting the enzyme 1. So when the enzyme 1 stops their works, enzyme 2 and enzyme 3 that will be also affected. So this is the feedback inhibition process. So what are the characteristics of these inhibitors? These are the chemicals that reduce the rate of enzymatic reaction. They are usually specific and they work at the lower concentration and they block the enzyme but they do not usually destroy it. And what are the examples for that? Many drugs, poison that is acting as the inhibitor for the nervous system and some other heavy metal that is also acting as the inhibitors. So there are some irreversible inhibitors that combine with the functional group of the amino acid in the active site irreversibly Now, what are the example? Nerve gases, pesticide, organophosphorus. These are the some of the examples. So this also shows the type of the inhibition, competitive and the non-competitive type of the inhibitions. So this is the example taken from the Krebs cycle succinate that is converting into the fumarate, and the succinate dehydrogenase enzyme that is catalyzing this type of the reaction. This is the example you can see succinate CH2COOH, CH2COOH, and fumarate CHCOOH. Uh, so another similar kind of the compound. This is the substrate actually. This is actually the substrate, and succinate dehydrogenase enzyme that is acting on that one. This is more similar to the succinate. This is the malonate actually. CH2COH, CH2COH. This these are almost the same. If this malonate, actually this succinate dehydrogenase that is meant for this succinate, and that is not meant for the malonate. But in the competitive inhibition process, this these have the similarity that actually bind with this enzyme, so there is no room for this substrate. succinate के लिए उसमें कोई room नहीं होगा when this malonate that binds with the succinate dehydrogenase enzyme. So this will not be doing its work. So non-competitive about cyanides, maybe, and some other metal ions, heavy metals, that is combining with the enzyme. So the active site that has been changed. And for that purposes, EDT and some other things that can be used to chelate these. So application of the inhibitors in negative feedback endpoint are end products, inhibition, poison, snake bite, plant alkaloids, nerve gases, medicine, antibiotics, sulfonamides, sedatives, and stimulants. These are the application of the inhibitors, how they can be work in these areas. So look at this one again. The feedback that shows the feedback inhibition process, different kind of the enzyme you can see here. These are the enzymes. Uh, First enzyme, second enzyme, third enzyme. So these are catalyzed. For example, any one of this one, the product that if that is inhibiting, for example, this F that is inhibiting the first enzyme. So it's mean later on the production of B is affected, production of C is affected, production of D is affected. So it's a feedback inhibition process. This is also called as the negative feedback inhibition process. In this case, this is acting as an inhibitor for the first enzyme. This one, first enzyme. So the end products are controlling their own rate of the production. Taking example from the cell level, phosphofructokinase and ATP. Phosphofructokinase, that is actually a product enzyme that is working in the glycolysis process, and at that step, uh, so look at this reaction. Fructose 6 phosphate is the substrate. Fructose 6 phosphate in the presence of the ATP that is converting into fructose 1,6 bisphosphate. And this reaction is catalyzed by phosphofructokinase actually. So look at this one. how that is acting as the inhibitor when the cell level of the ATP is higher. This higher ATP that is acting as the non-competitive inhibitor of the phosphofructokinase. So the cell stops utilizing the ATP and to maintain their energy level. Otherwise, the ATP that will be finished in the cell and the cell lack of the energy. As ATP is used as up the inhibition stops and the reaction speeds up again. The switch allosteric inhibition sometimes the inhibition process is of the type of the allosteric. Allosteric, as I have told you earlier in previous lectures, that allosteric means the other sides. At some other points, uh, this is the active site, and this is the allosteric site. So this is the substrate. This is the substrate that combined with that enzyme. So this is the allosteric point. This one, allosteric site. So this actually control the configuration of this active site. So when there is a attached molecule, the active site that can be changed. So that switch off the working of the enzymes. So enzymes inhibitor fits into the allosteric site, normally on this side, this one. So changing this configuration of the active sites. So now you can see the substrate that cannot be fit into the active site. Substrate cannot fit into the active site. So look at this one, all the process. This is the active site. Normally the substrate and enzyme that make a good complex here. But when the allosteric site is filled, as in this case, the active site that has been now changed and the substrate that cannot fit into that position. So change in shape, conformational change as we have seen here. There is a conformational change. Negative feedback mechanism we have discussed earlier. Phosphofructokinase we have discussed earlier. In this case, the allosteric site that is covered with the ATP molecule when the ATP that is high in the cell. So stops the functions of the phosphofructokinase PFK that stops its function when the ATP quantity is high into the cell because that is acting as the allosteric inhibitor. So that's all for this lecture.